other week on Thursdays at 3:30 p.m. If you want to look at the list of upcoming webinars, you can go to the CIBERNED webpage and you can check the program over there. I think the list of webinars is ready until the end of the year. So please feel free to go over there. Also, before I start, let me remind you that this webinar will be recorded, and as usually, it will be available at CIBERNED webpage in the upcoming days. I would kindly ask you, everyone, to keep your microphones and cameras off, so we have a nice streaming of the presentation. And also remind you, for those of you who haven't been part of this seminar series before, we have about 30 to 40 minutes presentation, and then we'll have some time for discussion of the seminar. So if you're interested in posting a questions to the speaker, please just raise your hand using the application, or just send a message stating your interest and your affiliation, and I will give you the floor in a first come, first serve basis. Okay, so having said that, it is now for me a pleasure to introduce you the today's speaker, Dr. Marta Cortés Cantelli. Marta is got her PhD in 2004 from the Autonomous University of Madrid, in a thesis focused on the role of transcription factor in neuronal differentiation and brain injury. In 2008, she moved to the Rockefeller University at New York to study the vascular component of Alzheimer's disease, where she was a postdoc until 2008, and then became a research associate till 2014. In 2015, she returned to Spain with a European Marie Curie Fellowship to start a line of research at the CENIC in Madrid, deciphering the heart-brain connection in Alzheimer's. In particular, the impact of cardiovascular risk factors and atherosclerosis in middle age on brain function, neurodegeneration, and neuroinflammation. She was later selected to be a Miguel Servet researcher, and in the last year, she actually has become an independent researcher at the Instituto de Investigación Sanitaria Fundación Jiménez Díaz in Madrid, to work on the research line that I just mentioned on the vascular component of Alzheimer's disease. So her talk today is actually titled Vascular Component of Alzheimer's Disease. And Marta, thank you very much for your willingness to share your work with us and for being here today. And please feel free to start with your presentation. Thank you.
Well, first of all, thank you very much. Let me share the PowerPoint. As I said, thank you very much for letting me explain our work studying the vascular component of Alzheimer's. I'm sure I don't have to get into many details about this disorder in this venue, but a few bullet points just to mention that it is a very complex and multifactorial neurodegenerative disease. There are 35 million people affected right now worldwide, and this number is predicted to reach 100 million people by 2050 if we don't find any therapeutic approaches. The main pathological hallmarks, besides this profound atrophy that you can see in the figure, the two main pathological hallmarks are the amyloid plaques and the tau tangles. And the treatments that are available right now are only symptomatic agents. So there are a lot of pharmacological and non-pharmacological approaches under development right now. This disorder was discovered by the German physician, Dr. Alois Alzheimer, more than a century ago. And when he wrote the first autopsy report of the first patient with Alzheimer's, whose name was Auguste de Dieter, he, of course, defined the plaques and the tangles, but he also wrote that he was seeing atherosclerosis of the small cerebral blood vessels. It is true that this vascular component has not been studied very in depth, but the truth is that the vasculature of the brain is in very close connection to the brain parenchyma. Here you can see a brain MRI. It's actually my MRI, it's my brain. And you can see the blood vessels in black and how they are in very close connection with the brain parenchyma. So it is not difficult to envision that if something is happening to one or more of those vessels, acutely or in a chronic fashion, it's going to affect the brain function. Here you can see a representation of the capillary or the brain vasculature, and it's actually very curious to know that the network of capillaries, if you put them all together, it's more than 500 kilometers. So I think it's something that we should have in mind when diagnosing and treating this disorder. Actually, there are some non-pharmacological therapeutic strategies for Alzheimer's. For example, it is known that different cardiovascular risk factors such as obesity, hypertension, smoking, and diabetes increase the risk for cognitive decline. And the other way around, physical activity and a Mediterranean diet, for example, decrease the risk. This has been reviewed by many authors. For example, Professor Yadecola in 2019 reviewed all the evidence linking this type of factors together with other lifestyle factors with the development of different types of dementia. You can see here in this summary table how factors such as smoking, physical activity, BMI, and hypertension, dyslipidemia, stroke, they are all related with, and there is established evidence that those factors are linked to the development of Alzheimer's and other types of dementia, actually. This has been even quantified, and it's been estimated that around a third of Alzheimer's disease cases worldwide might be attributable to these potentially modifiable risk factors. We are talking here about 10 million people. So, with different cardiovascular risk factors and other non-modifiable factors, such as, for example, age and ApoE4 genotype, are linked to increased risk for suffering cardiovascular disease and Alzheimer's. And it's also known that a high cognitive reserve, a healthy lifestyle, and the control of some of these cardiovascular risk factors may reduce the risk of suffering dementia, especially if this type of intervention is carried out in middle age. Besides this connection besides this sharing of cardiovascular risk factors by both disorders, there is also some other evidence linking the heart and the brain in Alzheimer's. It's known that there is atherosclerosis in the circle of Willis in Alzheimer's disease patients, but also in extracranial arteries, such as, for example, the carotids and the femoral arteries. And it's been even described the presence of A-beta in the atheroma plaques. There is also neurovascular unit dysfunction with all the different components of this structural tear, the endothelial cells, the pericytes, the tight junctions, the astrocytic end feet. There is blood-brain barrier leakiness in Alzheimer's. There is also the position of amyloid in the vessels known as cerebral amyloid angiopathy. And there is also fibrin clot formation, and I'll get into more detail about that in a second. Besides this, there is also microvascular pathology in AD, cerebral hypoperfusion, hypertensive vascular dysfunction and remodeling, arterial stiffness, and also intramyocardial A-beta deposits in the AD patients. So as I said before, I'm going to focus my talk in two different lines of research that I've been doing during the past years. One of them is analyzing the impact of these fibrin clots that are formed abnormally in Alzheimer's, and also the relationship between atherosclerosis and Alzheimer's in preclinical stages. So going with the first part, first of all, I need to show the coagulation cascade. It's a very complex cascade and a very complex process where different coagulation factors are induced one after the other, and some of them even repress the others when they are no longer needed. This process is tightly regulated, the thrombosis or the coagulation and the fibrinolysis. And the final step of the coagulation cascade is the formation of fibrinogen into fibrin by the action of thrombin. What is known in AD is that several of these coagulation factors are induced. Not only coagulation factors such as factor 12, factor 7, factor 5, actually thrombin itself, but also there is platelet activation, and there is increased levels of of the PI1 inhibitor. Also, there is decreased levels of the fibrinolytic components. So you can imagine that with increased levels of the coagulation part and decreased levels of the fibrinolytic system, we may think that there is a tendency in the AD brain of accumulating fibrin and fibrinogen. Well, this is something that we looked up while I was a postdoc at Rockefeller. We check different human AD brain samples, and we indeed observe fibrin and fibrinogen deposition in the brain parenchyma, but also in the brain vessels. You can see here in the vessels that some of them are completely occluded and some of them are only partially. It was interesting to see that these fibrin deposits were in very close connection to A-beta deposits. In order to quantify the amount of fibrin that we were having, we set up a protocol for fibrin extraction, and we received several samples from several patients from the Harvard Brain Tissue Resource Center. And as you can see here, we extracted the fibrin and we ran our Western blot from the insoluble fraction. You can see here the control patients, the age-matched control patients, don't have any fibrin in their brains. Fibrin is excluded by the blood-brain barrier in normal conditions. But you can see here the AD patients, a high proportion of them do present fibrin in the brains. Each lane corresponds to an individual AD patients. We calculated from 60 to 70% of them. We work in the lab with transgenic AD mouse models. The one that we use is the TGCRND8 mice that overexpress 2 different mutations in the APP gene. So we wanted also to see if this fibrin deposition or fibrinogen deposition was also happening in our AD mouse line. So immunohistochemistry show increased fibrinogen deposition also in areas where A-beta was present. We also extracted the whole amount of fibrin and ran it in a Western blot. You can see wild-type mice do not have any fibrin in their brains. AD mice do have a lot. And actually, well, this is the 6010 antibody against A-beta to confirm that they were indeed AD mice. So fibrin is the main protein component of blood clots. So if we see this abnormal deposition, maybe the fibrin clot formation or the thrombosis process itself is also altered in these mice. Well, we open a cranial window, an acute cranial window, and we went to the microscope and labeled fluorescently labeled the blood flow, and we applied ferric chloride to induce clot formation, as you can see in the video. This is a potent chemical that is going to induce fibrin clot formation very quickly, and after a second topical application of ferric chloride, the blood vessel is almost completely occluded, as you can see in the movie. We quantify this in different mice and in different vessels. And as you can see in the graph, AD mice clot faster and even before the addition of any ferric chloride compared to the wild-type mice. So thrombosis was also altered. What about without inducing any thrombosis? How does the vasculature look like? Well, we open, in this case, a chronic cranial window in collaboration with a lab at Cornell, and we quantify the amount of stalled brain capillaries that were not flowing in regular conditions. And we observed that there was an increased amount of capillaries that were actually blocked in AD mice. So that could be partially the reason why there is hypoperfusion in this pathology. Okay, so with this background in mind, we wonder if there is a procoagulant state in AD, maybe a therapeutic approach aimed at normalizing this thrombosis could be beneficial. Well, there were some evidences in the literature showing that patients, for example, with atrial fibrillation that undergo long-term anticoagulation, there were studies showing that these anticoagulants and in this type of patients, sorry, there is something in this type of patients was linked to a decreased risk of suffering dementia. There were also some other evidence in the literature in some AD mouse models and in a very small cohort of AD patients that the use of classic anticoagulants, such as, for example, heparin or vitamin K antagonists, such as curcumin, could be beneficial. However, the problem with these classic anticoagulants, and here we have again the coagulation cascade, is that they interact or they block the action of different coagulation factors at the same time. The classic anticoagulants are the levels on where they act are marked in yellow and in green, and you can see that they are affecting many different coagulation proteins, and one of the problems with that is that they are not very predictable, and also they are influenced by other proteins and by food, for example, since they are vitamin K antagonists. So and they have secondary effects such as hemorrhages that are very dangerous for fragile patients, such as, for example, the elderly or Alzheimer's disease patients. So that's why in the last decade or so, maybe more, these novel anticoagulants have been developed that target just one coagulation factor. They present less secondary effects. And we decided to focus on a direct thrombin inhibitor. It only inhibits thrombin. It doesn't interact with food. It presents very low binding to plasma proteins. It presents a very predictable pharmacokinetic profile, and the patients don't need routine coagulation monitoring. And also, and very important for us, is that it presents a strong efficacy and safety pattern in elderly subjects and decreased risk of bleeding, especially intracerebral hemorrhage. So we went ahead and decided to treat our mice with one of the direct thrombin inhibitors that are already approved for other indications, that is called dabigatran. So we use, as I said before, the TGCR-ND8 mouse model that starts the amyloid deposition at around 9 weeks. So we started the treatment right before that amyloid deposition starts. It was given in the chow, so it was very easy to give. We sacrificed the mice at 2 different time points, at 30 weeks of age or 60 weeks of age, and we extracted blood at the different time points as well, basically to make sure that the dabigatran plasma concentrations were as we expected they should be, and they actually were. One of the techniques that we used was arterial spin labeling MRI to measure cerebral perfusion. So, as I said before, these mice and the AD patients as well present cerebral hypoperfusion. In this case, it was around 20%. And what we observed with dabigatran-treated mice is that that cerebral perfusion was gone. Cerebral blood flow was actually preserved in AD mice treated long-term with dabigatran. Something else that we check in these mice was we did many immunohistochemistries, actually. One of them was to detect the amyloid oligomers with the antibody NAB61. Oligomers are one of the most toxic species of A beta. And as expected, we observe an increase, a significant increase in the amount of oligomers with age in the AD mice treated with placebo. However, the AD mice treated with dabigatran presented half the amount of oligomers after treatment. We also check for neuroinflammation. One of the markers that we used was CD68 that marks Stein's phagocytic microglia. You can see here the nice clusters of microglia around the plaques, and how it increases also with age. That was also expected in our AD mice. And that neuroinflammation was decreased by half with dabigatran treatment. Another marker that I wanted to show you was the astrocytic end-feed marker aquaporin-4, which checked for blood-brain barrier function. And we observed that aquaporin-4, this is a wild-type mouse. You can see how it nicely stains perivascular or shows a perivascular staining, very close to the endothelial cell marker. However, in the AD mice, that perivascular staining was shifted towards staining in the plaques. You can see it here. And the perivascular staining was quite decreased. We quantify this as well. And we observe a 30% decrease in the perivascular staining of aquaporin-4. And actually, that perivascular staining was almost recovered with dabigatran treatment. So long-term dabigatran treatment was able to ameliorate amyloid burden, neuroinflammation, and blood-brain barrier damage in AD. Last but not least, we also perform memory tasks. And I don't think I have to go into explaining what advanced maze is in this venue. So I'm just going to skip that part. If someone has any questions, I'd be happy to answer it at the end. But basically, we perform 7 days of training, and 24 hours after the last training day, we did a pro trial. And this is the videos I'm showing here are the ones for the pro trial. They are fast forward, of course. This is where the escape box was. This is a wild type. This is an AD. The mice are under those black boxes. You can see how the wild type remembers where the escape hole was, and the AD mouse doesn't remember. So we quantify this, and there was a significant difference between target and non-target quadrants in wild type, as expected. AD mice did not remember where the target quadrant was, or which one was the target quadrant or the target hole. However, the AD mice treated with dabigatran did remember perfectly where the escape hole was. So anticoagulation with dabigatran was able to prevent this memory decline. So as a summary from this first part, what we think is happening is that in an Alzheimer's disease scenario, there is fibrin clot formation in the blood vessels that is altering or affecting the cerebral blood flow. This is also having an impact on blood-brain barrier function and permeability. There is amyloid beta deposition that is going to also interact with the fibrin that is being deposited in the parenchyma. This is going to go along with astrogliosis, microgliosis, recruitment of T cells that I didn't have time to explain, but we also check for recruitment of T cells. And all of this is going to have an impact on neuronal viability. What's happening when you treat with dabigatran and AD mouse? Well, the blood clots are gone. I didn't show this here either, but we check for fibrin deposition and it's gone in these mice. So fibrin clots are gone. The blood-brain barrier is much better preserved. There is still some blood-brain permeability, and there is still A beta deposits, because we don't think this is the only thing happening, but we think it has a profound impact. There is also inflammation, recruitment of T cells, but the neurons are much healthier and happier than in the AD scenario. So this is the first part. The second part is whether a procoagulant is, if we have a procoagulant state in AD, maybe a diagnostic approach aimed at identifying this procoagulant state could be useful, especially because we observe that not all AD patients have the same amount of procoagulant state. In fact, there are some of them that don't have any. So you don't want to treat with anticoagulants or with any other treatment an AD patient that don't need that specific treatment. AD is a multifactorial disease. We should not forget that. And I think the one treatment for all in this disorder is not working. And individualized diagnostic and therapeutic strategies, I think, are the future. So with this in mind, and this part that I'm going to present now is not published. So it's something that we are working on right now. we are developing different molecular imaging probes to be able to in vivo visualize this thrombosis. The first probe that we are working with is the fibrin binding probe that was developed by Peter Caravan from the Mass General. Here you can see one of the original papers from Caravan where they induce clot formation in the carotids, and it's very clear. We got the probe and we performed some positive control experiments with inducing thrombosis in the femoral artery using ferric chloride. Here you can see how we performed the surgery, and it was very easy to see that fibrin, the FBP, was indeed getting incorporated into the fibrin clot. So the probe was working. And with this, we actually got a JP&D project to focus a little bit more on developing this probe. I am happy to be the coordinator and work with this amazing team of partners that include Manuel Desco from the Gregorio Marañón Hospital, and actually he's also part of the CIBER, with Dag Selling from Sweden and Susanne Kosatz from Germany. So basically, we aim to develop a probe to detect fibrin in AD, not only in the brain vessels, but also we are going to couple it as a Trojan horse with a transferrin receptor antibody to be able to translocate the probe inside the brain parenchyma and be able to detect the deposits, the fibrin deposits that are also inside. That will increase, with that we will be able to increase the signal actually that we get in the PET. So very preliminary and please confidential results from the fibrin binding probe. We label it with zirconium 89, that is a radioisotope with a long life, or well, relatively, we at least we are able to follow up the probe for 5 days in the nanopet. The biodistribution assays, which is basically done at the last day of the PET, showed no difference in the brain uptake of this probe between wild type and AD mice. However, when we check the different time points and the different regions, we observe a significant increase in the uptake of the FBP probe in the AD mice. You can see here at the level of the whole brain, but also at the level of different regions of interest. It was very interesting to see also that studies, or when we perform the analysis but voxel-wise, we also observe this increased uptake in different regions. In parallel with this, we are also using another probe against platelets that is called ThromboTag, and this is done in collaboration with María Ángeles Moro from CENIC and Fernando Herraz from the Instituto de Química Médica, who is also part of another CIBERS. In this case, we perform a positive control experiment using one of the stroke models that María Ángeles Moro has set up in her lab. And you can see here how the platelet probe incorporates into the area. So in this case, we use a different experimental approach. We use orthogonal chemistry, in which the two different parts of the molecule, the antibody is injected first, and then the nanoparticles that are labeled with the second part are injected later. With that, you give time to the antibody to distribute perfectly throughout the organism, and then you can use a radioisotope that has a shorter half-life. In this case, gallium, which is only 68 minutes of half-life. So that's why we couldn't follow up these mice for longer periods of time. However, as we saw before with the FBP, we were able to see just at 24 hours after the injection an increased uptake of this probe in the AD mice in the brain. This is by biodistribution assays. When we perform the neuroimaging analysis, we again observe an increased uptake of the ThromboTag in the whole brain analysis, but also in the different regions analyzed. And the voxel-wise analysis show up the same. So these are very interesting results, and we are expanding the groups and the ages, and we hope to get these published very soon. So the neuroimaging biomarkers are very important, but the blood biomarkers are one of the fields that are expanding right now, especially to diagnose the neurodegenerative disorders, and also because they are much probably cheaper to perform. You don't have to inject any radioactivity in the patient, and it's also very very easy to get. So that's why we decided to also try to develop or find a blood biomarker to identify this procoagulant state. And this is an expanded figure of what I showed you before from this paper, where we also check for different regions of other patients. And again, you can see how controls have no fibrin, but 80 patients do have fibrin, but not all of them again. So we really want to identify these ones to select them to, for example, go under anticoagulant treatment. So for that, we requested samples from the Banco de Tejidos de la Fundación 100, from the BT100. We obtained post-mortem tissue samples in order to quantify the amount of fibrin of the different patients. And also, we obtained from the same patients blood samples and also arterial spin labeling MRI perfusion images. And this is done in collaboration with Brian Strange and Alberto Rábano. And we are right now finalizing the first analysis that look promising and will probably expand the number of patients soon. Okay, and moving towards the end of the talk, and this is the third part that I wanted to talk to you about, is what's the relationship of this procoagulant state or what's the relationship of AD with other vascular disorders? Well, the connection of Alzheimer's and atherosclerosis has been shown and proven in symptomatic stages. These are AD patients, and this is their circle of Willis that is full of atheroma plaques. You can see in comparison with controls. This has been demonstrated, as I said, in clinical stages by different people, different researchers. So the truth is that both disorders share risk factors, as I said in the introduction, and they both present very long asymptomatic phases. We're talking about 15 to 20 years of asymptomatic phases. It is true that the prevalence of AD increases by 3 in those individuals with severe atherosclerosis. So maybe are these two diseases coexistent over several years before one of them becomes symptomatic? And which one is the cause and which one is the consequence? Well, for that, I proposed to include different brain studies in the PESA cohort in collaboration with Dr. Valentin Fuster and Dr. Borja Ibáñez, who also happens to be part of one CIBER Cardiovascular. And these, they both work at CENIC in this, and they are actually the principal investigators of the PESA study. So what is the PESA study? It's the progression of early subclinical atherosclerosis. It started in 2010 when more than 4,000 individuals were recruited. They were all middle-aged, from 40 to 50 years of age, all of them asymptomatic. One of the exclusion criteria was actually that, not having or having any cardiovascular disease. So all of them were healthy. They have been followed up for different visits. We are now in visit 4 here. And during all these visits, a thorough diet questionnaire, physical activity measurement, cardiovascular risk factors, psychosocial factors, and a very a comprehensive vascular imaging protocol, including 2D and 3D ultrasound in the carotid, in the femoral, and in the aorta, and also the determination of coronary or calcium in the coronary arteries to quantify the CAP score. So In visit 1, this healthy population, 63% of them already had some subclinical atherosclerosis in one of the territories. This was quite striking because it's a high amount of participants. We are talking about people that are 46 years in average, so they are pretty young. In visit 2, subclinical atherosclerosis had progressed in more than 40% of these participants. So what about the brain? Well, in visit 1, the participants with more subclinical atherosclerosis were invited to join or to undergo to join the advanced imaging cohort that, among other things, they underwent an FDG PET. For vascular inflammation, but also it was very interesting because glucose uptake in the brain, that is FDG is an analog of glucose, measures synaptic activity and cerebral metabolism. So reduced FDG uptake is considered a marker for cerebral hypometabolism and in general for brain's health. So why is this important? Well, because alterations in the FDG PET uptake has been shown in presymptomatic individuals and is considered an early biomarker of AD. So these images that were acquired in the advanced imaging cohort were analyzed in collaboration with the Barcelona Beta Brain Research Center, that is the research center of the Pascal Maragall Foundation in Barcelona, particularly with Dr. Juan Domingo Gispert, who happens to be also part of the CIBER BBN. So we analyzed whether there was a relationship or an association between cardiovascular risk factors, subclinical atherosclerosis, and brain metabolism. So the first piece of data that I wanted to show you is that there was a significant association between cardiovascular risk score, in this case measured by the Framingham risk score at 30 years, with the brain brain hypometabolism. So the more cardiovascular risk factors, the less brain metabolism. This was also very interesting to see through voxel-wise analysis that some of the regions mostly affected were the angular gyri and the temporal gyri. Among the cardiovascular risk factors that compose the Framingham risk score, hypertension or the presence of hypertension was the one mostly affecting the cerebral hypometabolism, meaning that those participants with hypertension were the ones with the lower cerebral metabolism. Again, the voxel-wise analysis were very interesting to see because different regions, such as, for example, the cingulate gyri, that is part of the limbic system, involved in emotion, memory storage, decision-making, was one of the most hypometabolic regions. Why these regions are so important? Well, because they are part of the Alzheimer's disease hypometabolic signature. One of the next things that we wanted to check was whether carotid plaque burden or plaque burden in general, but the results I'm showing you now for the sake of time is only the plaque in the carotids, was also associated, the more plaque in the carotid was associated with less brain metabolism. Again, the voxel-wise analysis showed that the cingulate gyri and the right precuneus were quite affected by this hypometabolism. Again, part of the AD hypometabolic signature. So this is the last slide of the third part of the results that I wanted to show you. And just the take-home message that I want you to take is that Alzheimer's disease is a complex neurodegenerative disorder. There are several hypotheses on the table. My feeling is that probably all of them are right, and all of them coexist at some degree in the different AD patients. Here, for example, is depicted the amyloid hypothesis, the tau hypothesis, the neuroinflammation hypothesis, the vascular hypothesis, but there are many others. So we, since Alzheimer's disease is a multifactorial disorder, we should focus on the development of individualized diagnostic and therapeutic strategies to target the different mechanisms contributing to pathology. And one of them is, of course, the vascular component. So thank you very much for listening, and thank you very much to my team, because everything I've presented is thank you to them. Thank you to the collaborators. I joined the Instituto de Investigación Sanitaria a couple of months ago, and I already have different collaborations with different departments, neurology and nephrology, for example, and of course, cardiology, and also genetics with Carmen Ayuso. Of course, the NIC and all the support, Dr. Fuster, Ibañez, Moro, and the whole Program 5, all the different collaborators, and of course the funding agencies that are with them. Without them, it will be impossible to perform all this research. So thank you very much.
All right, thank you very much, Marta, for our very interesting presentation. So the paper is now open for questions. So if anyone is interested in making questions to Marta, please use the raise hand symbol in your application, in your screen, or just send me a message through the chat stating your name and affiliation, and I will give you the opportunity to do the question yourself. There is no need for writing the question. Okay, so while we wait for the first question, let me just also remind once again everyone that the CIBERNED Seminar Series is held every other week on Thursdays at 3:30 p.m. and they are open to the public. So everyone, whether they belong to CIBERNED or not, they are more than welcome. We already have the program ready for the next few months till the end of the year. So if you're interested in looking at that program, just please go ahead to our webpage and check the list of upcoming webinars. All right, so any questions? I don't see any raised hands.
I've left them speechless.
Okay. No questions for Marta? Okay, so let me just break the ice by making a more general question, Marta. And this is a more general question regarding to various dementia types. You know, when you look at the brains of postmortem brains of demented people, it's quite clear from the talking to the neuropathologists that you have quite a mixture of pathologies in there, and it is quite common to find combination of Alzheimer's disease and what is called vascular dementia. So what's your take on, you know, the differences, if there are any, on the pathophysiology of these two entities?
So I'm not a neuropathologist, but I do think that sometimes, or well, we should ask this to a neuropathologist, but I think it's very difficult to find in the case of Alzheimer's, a pure Alzheimer's type. As you said, most of them are mixed with other pathologies, in particular vascular pathology. It is also true that I think it's quite difficult to see only a vascular dementia type with nothing else. So I think we are talking about a spectrum that is overlapping, and in some cases it will affect more, and in other cases it will affect less. Why we think this is affecting more Alzheimer's or has a more profound role on this disorder? And I haven't explained anything about that, but something that we were working on at Rockefeller was also there is a specific binding between amyloid beta and fibrinogen. They bind and they make the clots more difficult to degrade. So this is something specific to Alzheimer's. That's why we think in those that have a vascular component and develop Alzheimer's, probably that's having an effect on the onset and progression. And that's why we think should be taken into account more than in other maybe neurodegenerative disorders.
All right, thank you. Okay, any other questions from the audience? Don't be shy, please. I haven't received any message through the chat. I don't see any raised hands either. Let me check this other way, just in case. No questions? Okay, let me make a second question, Marta, if you allows me.
Sure.
And this is, I'm not sure whether you mentioned this, I may have missed it, but are there any sort of epidemiological studies on people being treated chronically with anticoagulants or procoagulants treatments, and how that affect the risk of developing Alzheimer's later on?
Yeah, we actually just wrote a review about that is about to be published. Yeah, I did mention it very briefly in one of the slides. There are these patients that have atrial fibrillation, that one of the treatments that are given to them is anticoagulants. And there are several epidemiological studies, well, many actually, epidemiological studies showing a decreased incidence of dementia. It was given like that, not specifically Alzheimer's, in those taking anticoagulants. Right now, in fact, many studies are comparing the incidence of dementia in warfarin treated versus oral anticoagulant treated, and there is all this evidence in the literature. It's true that the dementia that they report is a composite dementia. There are maybe one or two that only divide that composite dementia in the different types, and it's not very clear what's happening. I don't know if it's because of the number of subjects in each of the dementia types, or maybe it's not like the diagnosis, because it wasn't focused on those specific trials. It's not very clear if for Alzheimer's is better than, for example, for vascular cognitive impairment. But the composite dementia, and taking into account that 70% of dementia cases are AD, we could maybe infer that it may be plausible, this treatment.
All right, thank you again. Okay, so let me ask again, for anyone interested in making questions, you still have time. So please don't be shy to make questions. No raised hands. No questions coming from the chat. Okay. So last call for questions. So this is your last chance to make a question to Marta, although you can always contact her directly through the email.
Oh, someone wrote in the chat.
Oh, really? Okay, I'm having here. So yeah, I don't know who OCN is, but please, put your microphone on and go ahead and make your question yourself, please.
Maybe he can't or she can't.
Well, the questions I see from
I cannot use the microphone.
Okay, you cannot use the microphone. So which systemic biomarkers would you suggest to be used?
Well, that's one of, I don't know. I would guess that a coagulation profile, but it's true that we use some of the ones that one could think of that could be useful, or not useful. The ones that you could think of as coagulation markers. And they are not that altered. So I think we need to go into more detail of Edwinomics, and maybe also developing one of these ultrasensitive systems that are right now being developed for A-beta, GFAP, NFL, those markers were not able to be detected a few years ago in plasma, and now with these ultrasensitive platforms, you can. So I think that will be the way to go, to find something that is really altered and try to develop one of these new ultrasensitive platforms.
All right, thank you, Marta, and thanks for this mysterious person who wrote the question. So we have a raised hand finally from Monica Mir. So Monica,
Yeah.
please go ahead.
Hello, and thanks for your presentation. It's very interesting. Just a curiosity, because the endothelial cells in the brain are a little bit different from the other endothelials in the body. There is something that makes you think that should be more at the fibrin more attracted by the ones in the brain.
We don't know how is being transported fibrinogen inside the brain. That's a very good question, or that's one of the things that we really wanted to focus on in the next few years. I'm not sure if it's endothelial cells or, for example, there is the position of A-beta in the vessels as cerebral amyloid angiopathy. Since fibrinogen interacts with A-beta, that could be one of the ways to get stuck into the vessel and then maybe get transported inside. Because the truth is that there is something that is transporting fibrinogen inside, because there is a position intravascularly, but also in the parenchyma. So that's a great question. We don't know. We don't know. We don't know if it's the endothelial cells or which receptors in the endothelial cells are the responsible to that.
All right, thank you, Marta. Thank you, Monica. So any other questions? No more questions? I don't see any other raised hands or messages coming through the chat. No? Okay, so if there are no more questions, then I think it's time to close the session. But not before thanking Marta for her very interesting and inspiring presentation. And thank you all of you for attending as well. And I hope to see you in the next webinar in a couple of weeks. So have a great afternoon, everyone. And thank you again, Marta.
Thank you.
Bye-bye.
Thank you for the invitation. Thanks. Bye.